Hi, everybody. My name is Ed Davis. I'm Senior Application Scientist at GeneCopoeia Incorporated, located in Rockville, Maryland, just outside of Washington, D.C. I'd like to thank you all for joining us for today's webinar entitled Applications for CRISPR Cas9 Stable Cell Lines. We really appreciate it. Before I go into the main part of the seminar, I'd just like to go over a few practical matters. First, you may ask me questions at any time. But in order to do so, your microphones will be muted so you won't be able to speak vocally to me. Instead, you'll need to type your questions into the questions box located in your control panel. If you can't see your control panel, it might be hidden, in which case look for a small strip of buttons on one side of the screen. One of those buttons will be orange with a white arrow in it, and if you click on that button, you'll then be able to see the control panel and hence your questions box. Secondly, in order to keep the flow of the talk going smoothly, I'm going to wait until the end of the seminar to answer your questions, and I will do so in the order in which they were received. And when I do so, I will read both the question and the answer to the question out loud in order for everybody to hear it. And finally, I'll be sending you a couple of links, web links, to either download a technical note or register for other future webinars, and I'll be doing that through the chat box, which is also located in your control panel. But I'll be giving you a heads up before that. All right, so with that, I'm going to go ahead and get into the seminar. All right, so first I want to tell you who we are at GeneCopoeia and what we're about. We are a company that's focused primarily on products and services for functional genomics and cellular biology. And one of the main product lines that we offer, and in fact this goes back to our founding days in 1999, is plasmid DNA clones. And we started off by offering clones for open reading frame, or ORF. That is, we have large genome-wide collections of primarily human, mouse, and in some cases rat open reading frame clones. They're ready to use the day you get them. And they come in various configurations, either with or without fusion tags, different promoters, lentiviral and non-viral formats, etc. But we've since expanded out to include other clone types like promoter clones, microRNA clones of various types like 3' UTR target clones, precursors, and inhibitors. CRISPR clones, that is primarily for single guide RNA expressing clones. shRNA clones for more traditional knockdown experiments. And we also offer a number of solutions for viral systems, primarily lentivirus and much more recently adeno-associated virus, or AAV. In addition, we offer a large selection of kits and reagents for various applications, such as for transfection reagents. We have a lot of luciferase solutions, both traditional Firefly and Manila based, as well as Gosi luciferase. Fluorescence in situ hybridization, or FISH probes. Indel detection kits, which you'll hear about is related to CRISPR applications. We offer a number of cloning kits. And finally, we have a large selection of reagents for fluorescent detection, including cell function assays for like apoptosis or autophagy, nucleic acid detection kits for either measuring quantities of nucleic acids or detecting on gels, cell structure probes, so these are strains for things like organelles like Golgi and mitochondria, and also a number of fluorescent dyes for conjugating to various agents like antibodies. But of course, you're here to learn more about CRISPR, so let's get into that. All right, so again, this webinar is focused pretty specifically on Cas9 expressing stable cell lines. And I'll go into more about the detail later, but I just want to let you know that these are cell lines that contain stably expressing Cas9 nuclease, as shown here. And we have many pre-made cell lines. Currently, the number of cell lines that we have pre-made with Cas9 expression is 44. But we're also making more through time, or you can have us integrate Cas9 in your cell line for you. In addition, plasmids are available for do-it-yourself stable cell line creation. And these are ideal for single guide RNA library screening, validation, inducible CRISPR, and more. All right, so here's an outline of today's seminar. First, I'm going to go over genome editing basic technologies, so some background. Then I'm going to talk about applications, general applications for genome editing. Next, I'm going to talk about the 4 major applications for Cas9 stable cell lines that we discuss. These are sgRNA validation, lentiviral CRISPR, CRISPR library screening, and inducible CRISPR. All right, so let's go over genome editing technologies. So now most genome editing events by CRISPR, or as shown here, TALEN, which is an older technology, begin with a single event, which is a targeted double-strand break, or DSB. Okay, and that's because CRISPRs in their most commonly used form are endonucleases. They're also called RNA-guided endonucleases, or RGENs. So what they do is they come along and they find a specific sequence in a chromosome and they create a double-strand break at that site. And then this break is repaired usually by one of two pathways, or mechanisms actually. The first, shown on the right, is known as homologous recombination, also known as homology-directed repair. This is a fairly natural form of DNA repair. It's what occurs during a lot of DNA damage where you have a homolog or a sister chromatid present. It also occurs in meiosis when double-strand breaks occur. And what happens is you have the double-strand break is repaired by homologous recombination between regions surrounding the break that are identical or nearly identical or homologous. And so what happens is the donor, as it's called, functions to repair the break by copying the information from the donor to the recipient broken chromosome. And when you can also mimic this by adding what's known as a donor DNA, an exogenous DNA, which can be a plasmid or an oligonucleotide. And in so doing, you can virtually knock in just about any sequence you like, as long as it's between the homology arms. This can be a selectable marker. It can be a mutation. You can correct a mutation, for example. But you get a targeted knock-in using this homologous recombination pathway. On the other hand, in the absence of homology, and this is what often is the case in non-dividing cells, you have a process known as non-homologous end joining, which is simply sticking the broken ends of the chromosome back together. Now, compared with homologous recombination, which is a relatively error-free process or low-error process, non-homologous end joining is actually a very highly error-prone process. And so what often happens is you get small insertions or deletions, also known as indels form, which can, 2/3 of the time, at least in theory, can create frameshift alleles if they're targeted to protein coding regions that can be used for targeted gene knockout. Okay, so how do CRISPR RGENs work, RNA-guided nucleases work? Well, there are many, many CRISPR systems in bacteria, and many have been adopted recently for genome editing, but still the most widely used CRISPR system for genome editing applications comes from the bacterium Streptococcus pyogenes, which is the same bacterium that causes strep throat. And as shown in this figure, CRISPR endonucleases are a two-component system. The first is the Cas9 nuclease, represented by this yellow shape from this figure. Okay, and the second component is known as a single guide RNA, which is an artificial chimera of two different RNA molecules, one of which contains sequence-specific target information. Okay, so when you put this sequence shown in blue into this single guide RNA, if the complex can find this sequence in the genome upstream of an NGG or PAM site, it then binds to this site by Watson-Crick base pairing and creates a double-strand break, which can, of course, be repaired by either of the mechanisms that I talked about earlier. Now, So that means then that this is a highly useful and highly programmable system for targeting almost any gene in any cell. Now, CRISPR, I'm sure you've heard about, has taken the world of molecular biology and biomedicine by storm. It has become a very highly efficient and useful method for doing many kinds of genome modification applications, such as knocking out genes or correcting disease-causing mutations, for example. It does suffer one fairly large limitation, and that is with its specificity. And this was first demonstrated a few years ago in Keith Young's lab, where they found that they could mutate different positions in a single guide RNA and then test them against a GFP reporter that's been integrated into the genome. And when they found each of these bars represents efficiencies of indel formation with each position mutated in the guide RNA, what they found was that even with these mismatches, you can often get very high, sometimes even 100% editing of the target site. There's some variation depending on which guide RNA it is. Each of these series of bar graphs represents different guide RNAs. But the bottom line is that you can mutate CRISPR guide RNA sequences and still get pretty efficient gene editing. And in fact, for 4 of the 6 tested guide RNAs, they found evidence for off-target mutagenesis and that occurred anywhere from 5 to 125% of the intended targets. So this was initially a very scary result, but it turns out that in recent years, more sophisticated design algorithms for sgRNAs have been implemented that allow you to either predict and avoid single guide RNA off targets, or they allow you to pick highly efficient or efficacious single guide RNAs with also low off target activity. And so the off target has become less of a problem for CRISPR guide RNAs than was initially feared. However, a number of technologies have been developed to mitigate CRISPR off-targeting. One is the double nicking strategy. You may or may not have heard this, but there's a mutant of Cas9 known as the nickase. There's actually 2 of them, but one of them is known as the D10A nickase. And this requires 2 guide RNAs that are spaced on opposite sides of the opposite strands, flanking the intended break site. And what they found was that while this double nickase strategy works very well, it can give you efficiencies of indel formation that can be as high as wild-type Cas9, but off-targeting is dramatically reduced, anywhere from 50 to 1,500-fold, at least in this initial paper. However, this is a difficult strategy to use. It's highly context-dependent, so there's a lot of constraints on orientation of the guide RNAs relative to each other, as well as the spacing between them. And so it's not really in a lot of common use unless you need really high fidelity applications. Another technological development that has come along to mitigate the off-targeting issues of CRISPR has come from a number of Cas9 point mutants isolated by the groups of Feng Zhang and Keith Young. Shown here are results from both of those labs, where they isolated a number of Cas9 point mutants that destabilize their interaction with the single guide RNAs. And so they actually allow on-target editing that can be as high as the wild type, not always, but often as high. But then they found, when they examined predicted off-target sites, that the levels of off-targeting were reduced to background levels. And the same is true of the other labs' results. So this was actually the most promising development so far for high-fidelity CRISPR-mediated genome editing, because unlike the nickase, you can use one single guide RNA, so there are no longer any orientation or spacing constraints, and you can get quite high levels of high-fidelity genome editing. All right, so now that I've given you that basic background, let's talk about some of the applications you can do. And again, these will all come into play later with the Cas9 stable cell lines. So this table summarizes some of those applications that you can do. One very common application is gene knockout. This can be done either with a plasmid donor or by non-homologous end joining. And these are old results, but they still apply. There's also correction of disease mutations, such as what was been done originally in when a group cured a heritable cataract disease mutation in mice. There's engineered disease resistance. This was using zinc finger technology, but this can be done with genome editing. And the classic example is researchers are knocking out the CCR5 gene, which can potentially cure patients of HIV infection. There's forward mutagenesis screens. You'll hear about this using CRISPR single-guide RNA libraries. In-frame fusion tagging. So let's say you want to add a V5 tag or a GFP tag to a gene, you can do that. And also there's transgene knock-ins, such as knocking in a transgene to like the safe harbor locus in humans or mice. All right, so let's talk about non-homologous end joining. This is essentially what it looks like. This is a result from a GeneCopoeia project that we did for a customer in a cell line, where we were the project was to knock out the HDAC6 gene, histone deacetylase 6 gene, in a cell line. This is the wild type sequence, and then we have a number of E. coli subclones, and I won't go into too much detail, but this cell line that we used actually has 5 copies of the HDAC6 gene. Okay, so what we see in 2 different cell clones, we found that these cell clones had 5 distinct knockout alleles. As you can see here, 1, 2, 3, 4, and 5. And each of these creates a frame shift. So you would predict, and the frame shift is near the initiator ATG. And the same is true of clone 2. So you would predict that this gene would be knocked out by frame shifting and created either a truncated protein that would be degraded or by causing nonsense mediated decay. You can also do knockouts using a donor plasmid. This was done using TALEN, but it's also done using CRISPR. As shown in this example, you can define a mutation and then use a selectable marker in the gene as well as a reporter gene, if you like. And when you transfect this with the CRISPR system, or TALEN in this case, and you select for drug resistance, in this case puromycin resistance, you're then selecting for homologous recombination events, and then you can knock out the gene using a donor construct. Another application, commonly used application for genome editing, is to do mutagenesis via homologous recombination, and that can often be done using an oligonucleotide donor. So these are single-strand oligonucleotides that are range in anywhere from 80 to 200 base pairs in length. And what you can do with this is you can introduce these oligonucleotides along with your CRISPR system. Now, this example that's shown here is actually, it's related to mutagenesis. It's actually correcting a disease mutation back to wild type. The procedure is the same, but this group used this strategy in mice to cure heritable cataract disease in mice. So when they sequenced the progeny mice, they found that the mutation was corrected, and in fact those mice no longer had heritable cataract disease. So that's another big application for CRISPR. And also, in addition, there's you can do in-frame fusion tagging. I mentioned that. This is also derived from a GeneCopoeia experiment which we did for in transgenic mice for a customer, where you can, if you want to, say, put a GFP tag at the 3' end of a gene, you then transfect or inject your cells or animals with the CRISPR components of Cas9 single-guide RNA, as well as the homologous donor shown here, and then you screen for knock-ins of the GFP that precede the stop codon. And finally, this is less commonly used application, but One can also do targeted gene activation or repression. This takes advantage of a mutant of Cas9 that has no nuclease activity, which is also known as dCas9 or dead Cas9. When dCas9 is fused to an effector molecule, which can be a transcription repressor or transcription activator, these can be targeted to genes to turn them on or turn them off without changing the genetic code. So it's, at least in the case of repression, it's similar to RNAi, but of course, there's no RNAi equivalent of transcription activation. But this is also a very useful application for CRISPR. All right, so back to Cas9 expressing stable cell lines. So I went through all these characteristics. That's what we want to talk about in this talk. But the point I want to drive home is that these can theoretically be used for all of these applications. But I'm going to tell you about the applications that are most well suited toward using the Cas9 expressing stable cell lines. All right, so let's talk about that first application. So one is sgRNA validation. Now, why? So I posed this question: why do functional validation? Well, that's because we strongly recommend when you get CRISPR components from GeneCopoeia that you do functional validation. We sequence verify the sgRNA clones, but by default we don't do functional validation, and that's because if you're using these in cell lines, we just simply cannot guarantee that they will function in any given cell line in any given experimental setup. Now, so the functional validation, and again, so we recommend doing this because cell culture-based genome editing projects can take 3 or more months to complete, but not all CRISPR single-guide RNAs are created equal. So we recommend testing these first before undergoing long genome editing projects to avoid potentially wasting time and expense. And so we recommend using a cell culture-based strategy using the T7 endonuclease 1 assay. So the way the functional validation assay works is it's PCR-based, and what you do is when you transfect a cell line with your single-guide RNA, and then you do PCR using primers flanking the double-strand break site. And in most cases, you don't get 100% cleavage in a bulk population of cells. Instead, you'll have PCR products, some of which are mutant and some of which are unmodified. But then when you denature and reanneal these products, you'll then get a mixed population of homoduplex mutant DNA, homoduplex unmodified DNA, or heteroduplex DNA, which can be either mutant over wild type or one type of mutant over another type of mutant. Okay, and so these heteroduplexes then are substrates for an enzyme called T7 endonuclease 1. And so that is what's used in our functional validation product known as the Indel-check T7 endonuclease 1 system, which is a functional validation system just like what I described. And so when you treat cells with CRISPR or TALEN, you then get a double-strand break in the chromosome, and then the indel-containing chromosomes, or all the chromosomes, are subject to PCR using primers flanking the break. Okay, and so when you go through the PCR denaturation and reannealing step that I described in the previous slide, as shown here, you can then take these products and digest them with endonuclease 1. And when you see the presence of 3 bands, all of which are predicted size, one is uncut and the other 2 are cut at specific sites as predicted, when you see this type of result, then you know that CRISPR worked well in your system. And this lane is undigested and this lane is digested. Okay, so we have a complete system of reagents for the Indel-check T7 endonuclease 1 system that come with either the PCR reagents or the T7 endonuclease 1 reagents alone, or you can buy a complete system with both. And you can purchase this with or without target-specific primers. Now, I'm telling you all this because if this is again one of the key applications that we talk about for CRISPR Cas9 stable cell lines. Instead, if you had a large number of single-guide RNA constructs to validate, well, then you might want to it might be easier to do this in a cell line that's expressing Cas9 stably. So now let's talk about the next application, the key application for Cas9 stable cell lines, and that's lentiviral CRISPR. Okay, so GeneCopoeia, just like other researchers have done, have made lentiviral constructs that express CRISPR components. This we have a two-component system. One lentiviral construct expresses the single guide RNA under control of the U6 promoter. And we have separated that into a second plasmid that expresses only Cas9. And the reason we do this, that we don't have Cas9 and single guide RNA on the same plasmid, is because the presence of Cas9 causes lentiviral titers to drop by anywhere from 10 to 100-fold because it's a fairly large gene. And so we recommend instead either taking these Cas9 expressing plasmids by themselves and using them to establish a Cas9 expressing stable cell line, and then transducing with this CRISPR single guide RNA lentiviral construct separately, or you can do this yourself. So you get much better titer when you do it that way. You only have to transduce with the single guide RNA expressing lentivirus. All right, so now that I've talked about the lentiviral constructs, that's going to lead me into a related application, which is CRISPR library screening. Okay, so what are CRISPR single guide RNA libraries? Well, most CRISPR single guide RNA libraries are designed for knockout. There are also some activation libraries, but by far the most commonly used libraries are for knockout. Okay, and these have largely taken the place of RNA interference-based screens, which were used for many years for doing things like drug discovery, drug target validation, etc. Now, it's important to keep in mind, though, that there are key differences between CRISPR and RNA interference for knocking genes out, especially in expression screens, because it can help you interpret your results. They're both, of course, designed to change the expression level. Of course, that's in a downward direction if you're using it for gene ablation studies like this. RNAi, though, as you probably know, causes a knockdown phenotype. You're doing really well if you're getting 90% knockdown, but you still always have some gene product left over in most cases. And this can be a problem for interpreting results. On the other hand, CRISPR is designed to knock genes out. That's because it causes a permanent change in the genetic code. Like I said, you get double-strand breaks, which can lead to frameshift mutations. That is not the case, of course, for RNAi, which, as I'm sure you know, acts post-transcriptionally or post-translationally on the mRNA. Okay, so how are CRISPR single guide RNAs used? Well, they're often used in one of two general ways. One is to infect a cell line with a pooled single guide RNA lentiviral particles, and then you screen the plates for response or phenotype of interest. And then you can do PCR or sequence individual guide RNAs, or you can do next generation sequencing on a bulk population to look for either an enrichment or a depletion of particular single guide RNAs. These are done in bulk. That's also known as a dropout or screen. Also, some libraries, smaller libraries, can be used individually in 96 well plates, where you can infect a cell line in individual wells with the single guide RNA expressing lentiviruses, and then you screen your plates for a phenotype of interest. Now, there are many different types of libraries out there, both academic and commercial. One of the most widely used libraries comes from the laboratory of Feng Zhang at the Broad Institute. This is a paper that described their Generation 2 single guide RNA libraries. This is a whole genome library, okay? And they actually have two different systems. One is an all-in-one system, and it's lentiviral based, where both the single guide RNA and the Cas9 are expressed on the same construct. And they also have a dual vector system where they're separated, like they talked about before. And this is where the Cas9 stable cell lines importance comes in, because what they found and what others have shown is that the titer, the relative functional viral titer of libraries containing single guide RNA alone is actually at least is about tenfold higher than the libraries containing both the Cas9 and single guide RNA. So this can really make a difference in your screening assay if you're doing a single guide RNA, if you're doing a CRISPR library screen. So again, we strongly recommend first using the two-component system and first establishing Cas9 expressing stable cell lines, and then using those stable cell lines to infect your cells with your single guide RNA libraries. Now again, those are whole genome libraries. Here's another example of a whole genome library. This report came out last year. Now in this example, the authors actually transduced their cell line with lentiviruses from a whole genome library. And what they found in a bulk population of cells is that there was a mixed population of Cas9 positive and Cas9 negative cells, even though they were the entire pool was puromycin resistant. And so what they ended up doing was they actually ended up subcloning into making individual cell line clones that purely expressed Cas9 in every cell. And what they found is that they looked at different clones and they found a number that did express Cas9 and then a number that were negative for Cas9. And these are flow cytometry experiments, by the way. So because of results like this, we strongly recommend that you first establish a Cas9 stable cell line and then go forward with your single guide RNA library screens. Now, of course, again, you can either establish these cell lines yourself, or you can purchase from one of our many pre-made Cas9 stable cell lines. And just as a final note on the library screening, we also have single guide RNA libraries at GeneCopoeia. These are the two-component system. Cas9 is on a separate plasmid. And we have 7 different pre-made libraries for anywhere from 57 to 658 genes covered for a number of different gene groups and pathways of interest. And these are available either as bacteria, DNA, or lentiviral particles, and can be purchased either pooled or individually arrayed. And our libraries have a number of advantages over the other commercial libraries or the large-scale genome libraries, and that is ours are individually constructed and cultured in E. coli before pooling. This makes possible their use in pools or as individual sgRNAs. The pools are limited to 150 single guide RNAs, which ensures excellent representation of each single guide RNA. Each one of these is sequence validated, so that provides high quality for each sgRNA. And they're also small library sizes, so these library sizes are limited to usually only a few hundred genes instead of covering all 20,000 genes at once. So this reduces the time and cost of screening. And of course, again, that is a fantastic application for CRISPR-Cas9 stable cell lines. Okay, and the last application for CRISPR-Cas9 stable cell lines I want to discuss is inducible CRISPR. So everything I've talked about so far has been using constitutively expressed CRISPR, but there are some situations where you may feel the need to use an inducible system, such as you might want to wait until after treatment with a compound, for example, or at a particular point in differentiation to get a knockout or some other kind of phenotype. So the first group to show this was in a paper by Gonzalez et al. A few years ago, where they created a Tet inducible system. The Tet system is a system that uses Tet responsive element adapted from bacteriophage that allows you to make a Cas9 nuclease gene that's inducible by doxycycline treatment. So doxycycline, you may or may not know, is an analog of tetracycline. And so what this group did was they knocked in onto one chromosome Cas9 under the expression of a Tet responsive promoter, and on the other chromosome they knocked in a constitutively expressed Tet responsive element. And then they used that system, as shown here, in induced pluripotent stem cells to analyze knockout of particular genes at a specific stage of development. So the way it works is you have your Cas9, inducible Cas9 expressing cells. You treat them with doxycycline that turns on Cas9 expression, and then you transfect or transduce with single guide RNAs, and then you look for your phenotype. And so this is another ideal application for Cas9 stable cell lines. And so when they did was when they treated pancreatic progenitor cells, when they differentiated the iPSC cells to pancreatic progenitor cells in the absence of dox, and then at this stage they transfected the guide RNAs and added doxycycline. And then they found by using the T7 assay that most of the cells show single stage specific induction of indels at 5 different genes. So again, this is an application that would be very difficult to do if you're using a transient transfection system. So now I just want to talk about what we at GeneCopoeia offer as far as in terms of Cas9 stable cell lines. Again, as I mentioned, we have a total of 43 Cas9 stable cell lines. 39 of these are human cell lines, and they can be categorized into different tissue types. For example, we have a number of, and I'm not going to go into too much detail, but we have a number of lung cancer cell lines, as well as the cervical cancer cell line HeLa. We have a large number of breast cancer cell lines like MCF7. We have human liver cancer cell lines. And again, all these are expressing constitutive expressed Cas9. Stomach or gastric cancer, pancreatic cancer, colon cancer, cecum cancer, and bone marrow, or blood cancer like Jurkat cells. In addition, we have the human embryonic kidney cell line, HEC293. That's not a cancer cell line, but it's an immortalized cell line. And one of these cell line offerings expresses the high-fidelity Cas9 that I mentioned earlier. And we also have neuroblastoma. And finally, we have the mouse neuroblastoma cancer cell line Neuro2A with different selection markers. And finally, we have the rat glioma cell line C6. So as you can see, we have a very large selection of pre-made Cas9 expressing cell lines. Or again, if you were to find that none of these suit your needs, you could always order our plasmid reagents from us and create a Cas9 stable cell line, or you can have us do it for you. All right, so with that, I just want to summarize. I told you that CRISPR is a highly effective method for many applications, from knockout, knock-in, activation, and more. I told you that GeneCopoeia provides a large number of cell lines that stably express the CRISPR Cas9 nuclease. And the GeneCopoeia's CRISPR Cas9 expressing stable cell lines provide great utility for several genome editing applications, such as sgRNA functional validation, lentiviral CRISPR, single guide RNA library screening, and inducible CRISPR. All right, so now if you want to know more about this topic on Cas9 stable cell lines and applications for them, you can download a technical note that we published recently, this is the download link, but I don't expect you to memorize or write down this link. Instead, I will copy and paste it into your chat box. And I just sent that along to you as well. So if you're interested in downloading this technical note, go ahead and download that link. And next, I want to tell you about some upcoming webinars that we have. Now, this is not related to genome editing, but you may have the need to use viral systems for DNA delivery. So this webinar is entitled How to Choose Between Adeno-Associated Virus and Lentivirus for DNA Delivery. It will be next Wednesday, April 12th at noon Eastern Time. This is the registration link, but I will also copy and paste that link into your chat box. Okay, and I just sent that link along to you. So if you're interested in registering for this webinar, go ahead and click on that link. And if you don't see the link, you might need to scroll down. But you can register for it the same way you register for this one. And finally, we will have another CRISPR genome editing related webinar coming up next month called Applications for Safe Harbor Transgenesis in Genome Editing. This will take place on Wednesday, April 19th at noon. And again, this is the registration link, which I will send to you in the chat box. Okay, and I just sent that link along to you as well. And with that, I'm going to stop, and I'd like to thank you all for your attention. I really appreciate it, and I will now take questions. Okay, so first question is, can we get a copy of this presentation afterward? Yes, I will make the slides available as well as a recording of this webinar available on our website. You can go ahead and you'll get instructions for obtaining that in a follow-up email. Second question is, do you offer all-in-one Cas9 CRISPR, sorry, I'm having trouble reading this, Cas9 single-guide RNA plasmids? Yes, we do. I didn't talk about that. I usually talk about that in a general webinar. So you would just have to go to our website and check out our many CRISPR genome editing applications and products. Next question is, how leaky is your Tet inducible Tet induction system? Well, we actually don't have that. The Tet induction system I mentioned is not ours. It's from a published paper. We don't sell the Tet induction system, so I can't really comment on that. I imagine it would be. I know the Tet system can be leaky, so but again, I can't comment on that. Next question is, do you have Cal3 based stable cell line Cas9? No, we don't. We don't have that one, but again, we can make it. That's something we have done for customers in the past. We've made some of these stable cell lines by request. Okay, does anybody else have any questions? Oh, yes. Somebody said thank you, and I want to say you're welcome. Okay, so if there are no more questions, again, I want to thank you all very much for attending this webinar. And if you have any more questions, just give me a call, 1-866-360-9531, extension 227. You can email me at edavis@genecopoeia.com, or go ahead and visit us on the web to check out our many different CRISPR-related products and services at www.genecopoeia.com. And I just want to say thanks and have a great day.